Hi, my name is Justin Luong, a PhD student from the Environmental Studies Department at the University of California, Santa Cruz. Thank you for the STEM Village for having me today. Today, I'll be presenting my dissertation research titled Functional Traits Can Inform California Prairie Planting Success During Extreme Drought. You can follow me on Twitter at Justin C. Luong or my ResearchGate for research updates and access the paper when published. Thank you again for joining me today for my presentation. Ecological restoration is needed to meet increasingly ambitious conservation targets in future years, but it's expensive and can have unpredictable outcomes. Ecological filter theory predicts that plants have to first withstand abiotic filters, then biotic filters, to successfully establish and reproduce in new environments. Functional traits are plant characteristics which could be morphological, physiological, or phenological, and they respond to environmental variation and help plants survive. Traits can inform ecological restoration by helping to select plants with appropriate traits for changing climates. Phylogenetics can account for unmeasured traits and underlying evolutionary patterns. Traits and phylogenetics are sometimes related, which can further expand trait-based selection for ecological restoration beyond species with measured traits, as a phylogenetic signal indicates closely related species will have similar trait values. Given this information, our research questions were, do functional traits explain plant community difference between drought and ambient rainfall plots? Do traits or phylogenetics explain planting mortality risk of restoration? And are leaf traits phylogenetically conserved in this plant community? Questions. We set up experimental plots at Younger Lagoon Reserve in Santa Cruz, California, United States. We built rainout shelters that exclude 60% of rainfall, thereby simulating a 1 in 100 year drought based on local historic rainfall patterns. Shelters were built in summer 2015 with specifications from the International Drought Experiment, also known as DroughtNet. Plots were mowed to remove all standing biomass in January 2016, then planted with the 12 California native species displayed above. Plots all had non-native species removal twice in the first year and not again thereafter. We measured the survival of planted species and the traits listed above for all the planted natives and extant non-native species in our plots. We determined phylogenetic distances relative to plant communities for each species in each plot using previously published phylogenetic data. PD0 represents the nearest neighbor distance, and PD50 represents the median phylogenetic distance. We tested differences in plant communities using Bray-Curtis dissimilarity and permutational analysis of variance. We used canonical correspondence analysis to see if traits explained community difference. We used Cox proportional hazard models to model effects of traits on mortality risk. We used Pagdel's lambda to test for a phylogenetic trait signal. Overall, we found the answer was yes to all of our original research questions. Functional traits can explain some differences in plant communities caused by drought. Traits and phylogenetics help explain planting mortality risk, and certain leaf traits are phylogenetically conserved. In the upper right-hand corner, you can see that drought and ambient rainfall plots had significantly different plant communities. This is a non-metric multidimensional space plot, where each point on the plot represents a plant community on a plot in 2018 or 2019. The further apart 2 points are from each other, the more dissimilar plant communities were. Ellipses represent the average distribution of each plant community grouping, and abundance-weighted community trait values of leaf lobeness, specific leaf area, and leaf carbon-nitrogen ratios were significant in explaining the variance we found between the treatments. Directions of the arrows represent where the traits were the greatest, except for delta carbon-13, which was not a significant predictor. We found the traits underlying plant community difference were also important for explaining planting mortality risk. The figures in the bottom left-hand corner represent these findings, where if a point crosses the null axis, it's non-significant. Traits to the right of the axis indicate higher trait values are related to higher mortality risk, and higher trait values are associated with decreased risk to the left of the null axis. In our system, increased leaf lobeness and slower growth rates were associated with decreased mortality risk, regardless of treatment. Certain traits like delta carbon-13, a proxy for water use efficiency, were only related to decreased mortality on drought plots, whereas leaf carbon-nitrogen ratios were only related to decreased mortality on ambient rainfall plots. Species that were more related were less likely to experience mortality on drought plots, but not ambient rainfall plots. We found that carbon-nitrogen ratios, but no other trait we measured, exhibited a phylogenetic signal, indicating that species closely related are more likely to have similar carbon-nitrogen ratios. In conclusion, we confirm that ecological filter theory is a useful framework for assessing restoration plantings in our system. Our findings suggest that phylogenetics and leaf traits, especially leaf lobeness, are useful mechanisms to understand planting mortality risk, community difference caused by drought, and to help improve future restoration outcomes. Phylogenetic signal and traits related to mortality can further expand trait-based selection to include closely related species. With that, I would like to thank you again for joining me for my presentation, and have a great day.